All right, moving on to 15.3, applications of genetic engineering. Our goal again is to analyze how heredity and family history can impact personal health, and also analyze strategies for prevention, detection, and treatment of communicable and chronic diseases. The far side says, when imprinting studies go awry. Do you get it? I do. All right, our objectives are three of them today. Describe the benefits of genetic engineering as they relate to agriculture and industry. Explain how recombinant DNA technology can improve human health, and also summarize the process of DNA fingerprinting and explain its usefulness. All right, think about it. Have you ever eaten genetically modified food lately? Well, if you've eaten corn, potatoes, or soy products in any of your meals this week, their chances are close to 100% that you've eaten foods modified in some way by genetic engineering. The first objective is to describe the benefits of genetic engineering as they relate to agricultural and industry. All right, GM crops, genetically modified. Almost everything we eat and much of what we wear come from living organisms. Researchers have used genetic engineering to try to improve the products we get from plants and animals. Genetic modification could lead to better, less expensive, and even more nutritious food, as well as less harmful manufacturing processes. And since introduction in 1996, not that long ago, genetically modified plants have become an important component to our food supply. One genetic modification uses bacterial genes that produces a protein known as Bt toxin. This toxin, thank God, is harmless to humans and most other animals, but enzymes in the digestive systems of insects convert Bt to a form that kills the insect. Plants with the Bt gene do not have to be sprayed with pesticides. In addition, they produce higher yields of crops too. Other useful genetic modifications include resistance to herbicides, which are chemicals that destroy weeds, and also resistance to viral infections too. The chart below is a summary of the adoption of genetically modified crops from 1996 to 2007. As you can see, the modified traits shown in the graph include herbicide tolerance, HT, this is herbicide tolerance, and insect resistance, BT. Some transgenic plants may also produce foods that are resistant to rot and spoilage. And engineers are currently developing GM plants that may produce plastics for the manufacturing industry, which would be nice. They use oil so much. Transgenic animals are becoming more important also in our food supply too. About 30% of the milk in the U.S. markets comes from cows that have been injected with hormones made by recombinant DNA techniques to increase milk production. Pigs can be genetically modified to produce more lean meat or high levels of healthy omega-3 acids. Using growth hormone genes, scientists have developed transgenic salmon that grow much more quickly than the wild salmon. Something interesting here, scientists in Canada combined spider genes, right, with the cells of lactating goats, milk-giving goats. The goats began to produce silk along with their milk. The silk can be extracted from the milk and woven into a thread that can be used to create light, tough, and flexible material. Kind of freaky. Scientists are working to combine a gene for lysozyme, an antibacterial protein found in human tears and breast milk, into the DNA of goats too. Milk then from these goats may help prevent infections in young children who drink it. Researchers hope that cloning will enable them to make copies of transgenic animals, which would increase the food supply and could help also save endangered species. In 2008, the U.S. government approved the sale of meat and milk from cloned animals. Cloning technology could allow farmers to duplicate the best qualities of prized animals without the time and complications of traditional breeding. Our objective was to describe the benefits of genetic engineering as they relate to agriculture and industry. So ideally, genetic modification could lead to better, less expensive, and more nutritious food, as well as less harmful manufacturing processes too. Objective 2 is explain how recombinant DNA technology can improve human health. Controversial stuff, right? Golden rice is a GM plant that contains increased amounts of provitamin A, also known as beta-carotene, and nutrition that is essential for human health. 2 genes engineered into the rice genome help the grains produce and accumulate the beta-carotene. That's the yellowy color. Provitamin A deficiencies produce serious medical problems, including infant blindness. There is a hope that provitamin A-rich golden rice will help prevent these problems in countries that don't have sufficient nutrition. Other scientists are developing transgenic plants and animals that produce human antibodies to fight diseases. So in the future, transgenic animals may provide us with an ample supply of our own proteins. Several laboratories have engineered transgenic sheep and pigs that produce human proteins in their milk, making it easy to collect and then refine the proteins. Many of these proteins can be used in disease prevention. So treating diseases, transgenic animals are often used as test subjects in medical science. They can simulate human diseases in which defective genes play a role. Scientists use models based on these simulations to follow the onset and progression of diseases and to construct tests of new drugs that might be useful for treatment. This approach has been used to develop models for disorders like Alzheimer's disease and arthritis. Recombinant DNA technology can be used to make important proteins that could prolong and even save human lives. For example, human growth hormone, which is used to treat patients suffering from pituitary dwarfism, is now widely available because it's mass-produced by recombinant bacteria. Gene therapy. All right. Gene therapy is the process of changing a gene to treat a medical disease or disorder. In gene therapy, an absent or faulty gene is replaced by a normal working gene. This process allows the body to make the protein or enzyme it needs, which eliminates the cause of the disorder. Other products now made in genetically engineered bacteria includes insulin to treat diabetes, blood clotting factors for hemophiliacs, and potential cancer-fighting molecules such as interleukin-2 and interferon. To deliver therapeutic genes to target cells, researchers engineer a virus that cannot reproduce or cause any harm. The DNA containing the therapeutic gene is then inserted into a modified virus. All right, you got a little virus right here. In theory, the virus will insert the healthy gene into the target cell and correct the defect. But gene therapy can be risky. In 1999, 18-year-old Jesse Gelsinger volunteered for a gene therapy experiment designed to treat a genetic disorder of his liver. He suffered a massive reaction from the viruses used to carry genes into his liver cells, and he died a few days later. For gene therapy to become an accepted treatment, we need more reliable ways to inserting working genes and to ensure that the DNA used in the therapy does no harm. Genetic testing can be used to determine if 2 prospective parents are carrying the alleles for genetic disorders such as cystic fibrosis. Because the CF allele has slightly different DNA sequences from its normal counterpart, genetic tests use labeled DNA probes that can detect and distinguish the complementary base sequences found in the disease-causing alleles. Some genetic tests search for changes in cutting sites of restriction enzymes, while others use PCR to detect differences between the lengths of normal and abnormal alleles. Genetic tests are now available for diagnosing hundreds of disorders. I apologize for the clarity of our visuals today. The same genes are not very active in every cell. By studying which genes are active and which are inactive in different cells, scientists can understand how the cells function normally and what happens when genes don't work as they should. Scientists use DNA microarray technology to study hundreds or even thousands of genes at once to understand their activity levels. A DNA microarray is a glass slide or silicon chip to which spots of a single-stranded DNA have been tightly attached. Typically, each spot contains a different DNA fragment, okay? See the DNA's in each little spot right there. Differently colored tags are used then to label the source of the DNA. So, for example, to compare the genes of cancer cells with the genes in normal cells, the messenger RNA would first be isolated from both types of cells. Each enzymes are used to copy the messenger RNA base sequence into single-stranded DNA labeled with fluorescent colors, as you can see in step B here. Red for the cancer cell and green for the normal cells. Both samples of labeled DNA would be mixed together, and then they would compete to bind to the complementary DNA sequence already in the microarray. Here we go, sorry. All right. Compete. Both samples, let me go back here, of labeled DNA will be mixed together. I already said that part. If the cancer cells produces more particular form of messenger RNA, then more red label molecules will bind at the spot for that gene, turning it red. Where a normal cell produces more RNA for another gene, this spot will then be green. Where there is no difference between the two cell types, this spot will be yellow because it contains both colors. This is a microarray. Green, red, and then yellows. So objective 2 was explain how recombinant DNA technology can improve human health. Today, recombinant DNA technology is a source of some of the most important and exciting advances in prevention and treatment of diseases, as we discussed. Objective 3, summarize the process of DNA fingerprinting and explain its uses. All right. So you should know already, no individual is exactly like any other genetically, except for identical twins who share the same genome. Chromosomes contain many regions with repeated DNA sequences that do not code for proteins. These vary from person to person. Here, one sample has 12 repeats between A and B, while the second has 9 repeats between the same genes, 12 and then 9 repeats. DNA fingerprinting can be used to identify individuals by analyzing these sections of DNA that may have little or no function, but that vary widely from one individual to another. In DNA fingerprinting, restriction enzymes first cut a small sample of human DNA into fragments. Containing genes and repeats. Note that the repeat fragments from these two samples are of different lengths. Next, gel electrophoresis separates the restriction fragments by size. Remember, from small back to large then. The DNA probe then detects the fragments that have highly variable regions, revealing a series of variously sized DNA bands. If enough recombinations of enzymes and probes are used, the resulting patterns of bands can be distinguished statistically, from that of any other individual in the world. DNA samples can be obtained from blood, sperm, or tissue, even from a hair strand if it has tissue at the root. The precision and reliability of DNA fingerprinting has revolutionized forensics, the scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn wrongful convictions. To date, DNA evidence has saved more than 110 wrongfully convicted prisoners from death sentences. DNA forensics is used in wildlife conservation as well. African elephants are a highly vulnerable species. Poachers who slaughter the animals mainly for their precious tusk have reduced their population dramatically. To stop the ivory trade, African officials now use DNA fingerprinting to identify the herds from which black market ivory has been taken. So hopefully they can find the people. So when genes are passed from parent to child, genetic recombination scrambles the molecular markers used for DNA fingerprinting, so ancestries can be difficult to trace. The Y chromosome, however, never undergoes crossing over, and only males carry it. Therefore, the Y chromosome passed directly from father to son with very few changes. Y chromosome analysis helps researchers settle long-standing historical questions. One such question was, did President Thomas Jefferson father the child of a slave? May have been answered in 1998. DNA testing showed that descendants of the son of Sally Hemings, a slave in Jefferson's Virginia state, carried his Y chromosome. This suggested Jefferson was the child's father, although the Thomas Jefferson Foundation continues to challenge that conclusion. He should just get real. Similarly, the small DNA molecules found in the mitochondria are passed with very few changes from mother to child in the cytoplasm of the egg cell. Because mitochondrial DNA, mtDNA, is directly passed from mother to child, your mtDNA is the same as your mother's mtDNA, which is the same as her mother's mtDNA. This means that if two people have an exact match in their mtDNA, mitochondrial DNA, then there's a very good chance that they share a common maternal ancestor. Objective 3 is to summarize the process of DNA fingerprinting and explain its uses. So DNA fingerprinting analyzes sections of DNA that may have little or no functions, but that vary widely from one individual to another. Our objectives, again, is to describe the benefits of genetic engineering as it relates to agriculture and industry, explain how recombinant DNA technology can improve human health, and also summarize the process of DNA fingerprinting and explain its uses. All these objectives help support our goal is to analyze heredity and family history can impact personal health, and analyze strategies for prevention, detection, and treatment of communicable and chronic diseases. Okay, we'll talk about this in class, guys. And there is my cute lizard picture. Ooh.